Hi guys, my name is Sarah Proventh. Today we're going to be talking about chemoreception and how that relates to the senses of both taste and smell. So what exactly are chemoreceptors? So chemoreceptors are actually receptors that can detect dissolved chemicals. Chemoreception is considered to be one of our oldest senses. Unicellular bacteria do use it to sense around their environment to see if it's okay for them to be around or not. Primitive animals also do use it to locate food and possible mates. We humans use it quite a bit more than we actually care to admit, and we won't truly know how important it is until we lose it. Usually, I'm sure most of you guys can relate to that when you guys catch a cold or something, can't smell or taste nothing, and it is not the best thing at all. So again, like I said before, we are going to be covering the sense of taste and the sense of smell. So let's first look at the sense of taste. So our tongue is covered in taste chemoreceptors. How do we know that? One taste bud is composed of 50 to 150 taste receptor cells. And along with those taste receptor cells, there are also support cells and regenerative basal cells. These taste receptors are not just consolidated to our tongue and clusters on our taste buds. It's also scattered throughout other regions of our mouth as well. For example, they are also located on our palate. So research actually shows that taste is a combination of 5 different sensations that we do encounter almost on a daily basis. Sweetness, sourness or acidity, saltiness, bitterness, and umami. So umami is a basic taste. It doesn't really taste like anything, but it enhances the flavors of food. It usually is associated with something called glutamate or other nucleotides. Taste buds do have 4 different cell types. So type 1 is glia-like support cells, which usually do perceive saltiness. Type 2 cells are referred to as receptor cells, and they perceive sweetness, bitterness, and umami. So sweetness and umami tastes are usually associated with receptors called T1R receptors. They usually have to have a different combinations of subunits for them to be perceived. Bitterness is usually perceived by T2R receptors. There's also type 3 cells, which are considered to be presynaptic cells. They usually perceive sourness. So the type 3 receptor cells usually perceive hydrogen ions, which is what makes something taste sour. There is also type 4 cells, which are called basal cells. Each sensation that we do have has a associated physiological process. So again, like I said, sourness, the perception of sourness is usually triggered by hydrogen ions. Saltiness is triggered by sodium ions. Sweetness and umami and bitterness are usually triggered by organic compounds. Our brain usually associates sweetness and umami with nutritious food, while bitterness is usually perceived as something toxic. That's why your first reaction when you taste something bitter is to spit it out, which makes sense. You don't want to ingest something that's going to harm you. It's one of the reflexes that our body has. So the taste pathway begins when the substance that we're going to be tasting is dissolved in the saliva and mucus of the mouth. Once that substance is dissolved, the ligands begin to interact with the apical membrane protein, which is on your taste receptor cells. As you can see, the taste receptor cell is right here. This is the pore that the substance is going to come through. This interaction that happens will then initiate a signal transduction cascade. So at the end of this cascade, chemical messenger molecules are released from the taste receptor cells. Then these chemical signals that have been released activate gustatory neurons. So after that, these neurons actually have axons that go through our cranial nerves to finally synapse on the medulla in our brain. So after they synapse on the medulla, the information is passed through the thalamus to the gustatory cortex. So this cortex is actually located inside a deep cortical region, which is called the insula. So the insula is what's highlighted in green right here. So that's where this information goes. And after that, it is interpreted based on which group of neurons is actually responding the strongest. These signals that travel to the insula can also initiate behavioral responses, such as feeding or feed-forward feedback. So yeah. Next, we are going to move on to the sense of smell. So the sense of smell can also be referred to as olfaction. Olfaction allows us to discriminate among millions of different odors or different smells. Everything that we smell gets processed in our brain, and then we eventually perceive it as something good or something bad. The olfactory bulb is an extension of our forebrain that actually gets information from our primary olfactory neurons. So how does this actually work? So how does something go from the outside environment to being perceived in our heads? Chemicals are dissolved in the olfactory epithelium, which is inside our nose. From there, the molecules then bind to olfactory receptors. So after they bind to the receptors, these molecules will initiate an electrical signal that goes from the sensory neurons to the olfactory bulb, which again is an extension of our forebrain. The olfactory bulb is actually responsible for relaying electrical signals to other parts of the brain so that they can be processed correctly. These signals that go to the other areas of the brain actually travel through something called the olfactory tract. The olfactory tract will take some of these signals and go to the olfactory cortex, which is a collection of neurons that are right behind the olfactory bulb, and the olfactory bulb will then identify what we are smelling. Now, in taste perception, the information usually goes to the thalamus. Well, in this case, the olfactory tract actually bypasses the thalamus and goes straight to the olfactory cortex. One of the really interesting things that you can note from our sense of smell is that ascending pathways from the olfactory bulb actually lead to the amygdala and the hippocampus. So the amygdala is responsible for our perception of emotions, and the hippocampus is responsible for our perception of memories. This would explain exactly why when you smell something in particular, it can bring up a specific memory or a specific feeling, whether it be good or bad. So how can we discriminate between different smells? How can we tell between this smell and that smell perfectly? I mean, we smell millions of things a day, so how can we tell the difference between the smell of, say, flowers and the smell of fresh bread? Research actually shows that each individual sensory neuron has a single type of olfactory receptor that will respond to only a very limited range of molecules. Cells that have the same receptors have axons that converge onto a couple secondary neurons in the olfactory bulb, which then will modify the information before sending it to the olfactory cortex. So that is how we can differentiate between different smells. I hope you guys enjoyed today's presentation. I look forward to seeing you guys in the next video.